Well, thank you very much for having me tonight to talk to you about this project that has been run as a collaboration between the university, the NIHR, across the NHS with the participation of the genetics lab and of course lots of consultants across the hospital here as well and Illumina. So I'm going to talk to you about today about a project called Next Generation Children Project, which was set to provide rapid genome sequencing for critically ill children. So genomic applications in intensive care units have been generating lots of interest over the last couple of years since we started our project. And because of rapid technological progress, it seems evident that this would be beneficial to this cohort. However, the evidence-based clinical utility of that kind of testing is still lacking. And there isn't a lot of data about health outcomes after that kind of testing. So some of the challenges for this genomic application includes the logistics of setting up a rapid pipeline, the cohort itself, which is highly heterogeneous, and the choice of the best genomic strategy. So the Next Generation Children Project is, as I said, a translational research project in collaboration with the NHS, University of Cambridge, NIHR, and Illumina, where patients receive whole genome sequencing. So the families are recruited as trio in ward upon pediatric consultant request, and the families consent for testing to find an immediate diagnosis in the child and for longer-term research as well. So the samples are processed in the genetics diagnostic lab here in Adam Brooks, and they are processed through a bespoke rapid pipeline that is integrated within the genetics laboratory. The DNA is shipped to Illumina twice a week by courier, and the data is then returned to the university for filtering and analysis of the genetic data. Then the pertinent findings are discussed weekly at multidisciplinary meeting with clinicians, research, and diagnostic scientists. And positive findings are confirmed urgently in the NHS, and a diagnostic report is issued as soon as possible to the clinicians. So my first question, and I'll ask a little bit the same question at the end of the presentation, is do you think that all patients that are offered whole genome sequencing in the future should sign a research consent? Still going? 146, 147, 48, yes? I think we're done? Yes. That's interesting. Very good. So as part of the national plans, the whole genome sequencing will be implemented through the NHS, and there are plans to integrate research a lot more than it's the case at the moment. So I'll be talking a little bit about the testing strategy that was chosen for this project, and that involves sequencing the genome of the child and both parents, if available, which is called trio sequencing. So the principle of trio sequencing relies on the comparison between the three genomes to reveal a genetic cause of the clinical diagnosis in the child. So the comparison may reveal a novel variant in the child that is not in the parents, and that's called a de novo finding, and that would be expected to be of dominant inheritance. Alternatively, parents may be carrier of a recessive variant in the same gene, which combined in the child would cause a genetic disorder. So the trio sequencing approach means that the information about inheritance is readily available to make decisions about the clinical significance of a genetic finding, and accelerates the interpretation of the findings. So the combination of this powerful inheritance filter together with searching for only very rare events predicted to have high impact on gene function is used to try to identify a likely causative variant in the child. So this approach is safe as it avoids most unexpected findings. It's rapid because the bioinformatics filter limits the number of results to analyze, and is powerful as all genes are analyzed at the same time. The main limitation of the approach is that it may be difficult to get parental samples, and also that some conditions with variable penetrance and dominant inheritance may be missed. So this slide shows a summary of the recruitment as of October last year, after 2 years of running this project. And the recruitment was primarily in NICU and in PICU, and through the urgent pediatric neurology clinic and genetics outpatient. So a number of these patients were also, of course, jointly managed through all these 3 specialties. So the large majority of, as you can see there, the large majority of these family was recruited as trio, and this represents the largest cohort of ICU trio patients so far. The diagnosis for each group on this part of the slide is represented in pink, and was in ICU from 15 to 17% in younger infants, to about 25% when children are older or recruited via pediatric neurology or genetics clinic, but of course in smaller numbers that you can see. And there were up to 50% in this cohort for older children, reflecting a larger phenotypic expression in older children and increased selection outside the intensive care settings. So the molecular diagnoses in this cohort so far were very broad and represented here proportional to how many times they were made. For example, there were 2 cases of Coffin-Series syndrome and 6 cases of epileptic encephalopathy. So most diagnoses were made only once, reflecting the heterogeneity of the cohort and the rarity of the disorders. And to give an example, in one case it was so rare that the Gene Disease Association was not even published yet. And thanks to data sharing initiatives, we were able to add our case to the first 3 ever cases to a manuscript that was under review. So we are currently running at over 285 families that have been recruited to this project, and at about 20% diagnostic rate for this cohort. So only findings thought to be the cause of the current clinical phenotype of the children were reported, although the absence of fully expressed phenotype in the neonatal period, or the limited knowledge, makes the interpretation of the findings quite complex. So some of these, interestingly, some of these disorders were strongly suspected based on the phenotype, but actually for most of them, it wasn't, which is demonstrating the utility of a wide spectrum approach, which is whole genome sequencing. So this is a summary of auditing the impact on clinical management for this family. The families recruited so far, that showed some broad categories of impact related to the impact on the clinical care pathway and treatment, the recurrence risk, and supportive or palliative care in case of fatal diagnosis. So as you can see on this slide, the major impact could be seen in the first category, where specialized care was initiated in younger children or informed in older children, while, interestingly, contrary to some predictions made in the literature, the impact on treatment modification and withdrawal was lower in this cohort so far. So, as a comparison, a classical pathway to diagnose a critically ill children may take months due to the technical limitation of the testing strategies and the iterative nature of the testing. So in contrast, in a rapid pipeline like the NGC project, the main difference contributing to the speed of diagnosis include the fact that all genes are tested at once, and most of all the genetic abnormalities that can be found can be tested by this one single technique. Also, the trio facilitates the interpretation, so less variants of uncertain significance are found, and there's no need for repeat testing for inheritance testing. So this means less weight and anxiety for patients and less appointments, and results in days versus months. And the little picture next in the diagram show potentially the number of appointments that patients have to go through to get a diagnosis. So last but not least, if there is no finding with that kind of approach, cyclic reanalysis and longer-term research and use of data sharing initiative may still allow for a diagnosis. So what does a negative result mean? Of course, the clinical diagnosis may not be of genetic etiology, but sometimes it can also mean that there is no one single gene that is responsible for the phenotype. It could also mean that the data is incomplete due to technical issues during sequencing or bioinformatics analysis. It could be also due to the fact that there isn't enough information about the patient phenotype to interpret a finding that is already there, or that there isn't enough information available about the finding itself to be able to assign it as causative. And sometimes the gene-disease association is not yet known. So this is my second question. So would you sign up for research if you were offered whole genome sequencing as part of a diagnosis workup for yourself or your family? How much did we have? We had quite a high score at the first question. Yes. 89%. I think it's increased. So this is a good sign that my talk was convincing. So I just wanted to show you, in summary, from our experience running the NGC project for the last 2 years, that some of the lessons that we've learned, and we have shown that rare genetic diseases are common in critically ill children, and that in very young children, the non-specificity of the clinical features mean that predicting which gene is affected is difficult. Therefore, 3-year whole genome sequencing, as we employed here, as a wide-spectrum tool, is the quickest and most powerful method to detect causative genetic variants in this cohort, which is showing the power of the translational research framework to increase the chances of diagnosis in this cohort of children. And to finish, this as a metaphor for the state of our current knowledge about rare diseases in critically ill children, and how the NGC project has shown that a wide exploratory genomic testing approach with close links with research is the most clinically useful model to diagnose rare diseases in critically ill children. And so I would like to acknowledge a very large number of people, of course the NGC families, the pediatrician, neonatologists, neurologists, geneticists who referred the patients, the university NIHR team, and in particular Lucy Raymond, who's who's a PI on this project, and with David Warwick, and the genetics lab team, the NHS team, and of course the Department of Clinical Genetics. This is all those contributions which made this project possible. Thank you very much.